Welcome again to our first virtual wega breakfast. So usually we do that in a nice location in Basel and have actual breakfast. So today I hope you brought something to some coffee to eat and drink, because we will have it just virtually. But I hope that next time we can again meet in person. So
Christian, you organized the delivery of croissant to everybody, I think. No, that's what you said.
Okay. So my name is Christian Wattinger, and I work for wega, leading our informatics solutions group. So I hope, let me share the screen quickly, sorry. So, and yes, first to very welcome again. And let me first quickly mention that you stay on mute during the presentations and feel free to ask questions in the meeting chat. So we will take them up later and the speakers will then answer them in the Q&A part afterwards. Now, I first would like to say a few words about wega. Our key mission is really to build the bridge between business and IT. Our strength are our employees, many of whom have dual qualifications in the life sciences and also the computer science, and a lot of experience at this interface. This enables us to understand the requirements and processes of our customers very well and to efficiently propose and successfully implement solutions. We offer actually 360 degrees informatics services in the life science and pharmaceutical sector with our 4 pillars of competence. We can provide the full spectrum of services, including business and informatics services, from the development and support of software to consulting in laboratory and research or clinical development, as well as validation and quality assurance. What our customers value about us is our sustainable long-term relationship with them, as well as our very stable employee workforce, and that we are backing this up with strong management commitment towards our clients. This has made us successful in the market for over 25 years, and we have grown by over 50% in the last 3 years. Recently, we see more and more interest around the topic of FAIR data, and many of our customers are doing projects in this area. And that's why we think it makes for a very interesting breakfast event today. So let me quickly highlight a few upcoming events. So one is the Future Labs Live, which will be in September. It will be also digital, like this event here, and it will be live from Basel. So we had hoped to really be there in physically, but I think it will be also virtual, like many of the events these days. So we'll have a talk there on digitalization of the lab requires data and connectivity standards. And then even more in the future, in October, we're still not sure if it will be physical or virtual. There will be the Analytica, which is a huge event where we have a booth and many talks there. And of course, you can find more events with wega involvement on our website. Now for breakfast, as I mentioned, we didn't organize the delivery, unfortunately, but we will do the delivery of some spicy brain food, I would say, thanks to our 2 speakers. And at this point, let me hand over to Daniel, who will say a quick word about FAIR and also about our speakers. Daniel.
Yes, thank you. No, I don't want to spend too much time, just to recall, because we have the chance to have, and we're very grateful to them for accepting to speak today. We have the chance to have 2 very experienced speakers, one from industry, another one from academia. Just a recall, but if you're there, you know about it. What FAIR means, so findable, accessible, interoperable, and reusable. of course, findable and accessible is one thing. Interoperable and reusable is much more useful in this context. Next slide, Christian. Thank you. Yeah, as I was saying, already getting the F out of FAIR is something of great value, and it's definitely not so straightforward. And of course, the more you can get about the three other letters, the better. So I just put a tweet here with an estimate of what could be the return on investment. But okay, of course, it can always be arguable. But in any case, there is value in that. And then how far you go into FAIR is always a question of cost and versus benefits. So as I said, we're very proud to have these two speakers today who went through the experience of trying to make the data fair. So the first one, well, just also one note. So the questions we'll put them all at the end after the two speakers, if you don't mind. So first, and please don't ask questions on the way you use the chat. So the first speaker will be Ewa. She's working at Roche as an IT business partner, and of course, I've gone through this wonderful exercise of trying to make the clinical data fair, and she'll talk about her experience around that, and then Professor Patrick Ruch. He has several hats, one of which is group leader and member of the SIB, Swiss Institute for Bioinformatics, where he leads the Elixir data platform. He's also head of research at the HESSO in Geneva, and he's a board member of Biomed IT. Now okay, thanks again, Ewa and Patrick, and with this I hand over to Ewa.
Thank you, Daniel, for the introduction. Let me share my screen, but you have to allow me to share the screen. I don't have this option.
Oh. So we tried that before. Oops.
Worked when we were testing it a couple of days ago.
Yes.
Now.
Sorry, it's just slow for me.
I have the option now. Perfect. Thank you very much. Let me know when you see my screen.
Yes, it's showing. Thanks.
Okay, so thank you very much for the invitation and for the introduction. As it was mentioned, I'm an IT business partner at Roche in PD Informatics Scientific Decision Support Network. And I'm supporting since already 10 years PD Biometrics, but also various initiatives around data sharing, data integration, and verification of the data. So everything that I will be sharing with you today is based on my experience in that field and in a large multi-year program that we are running at the moment, where I was involved in many initiatives on both business and IT side. And everything that we are trying to do as part of these initiatives is supposed to be sustainable and living model. We are trying to make sure that it can be widely followed without additional hurdles to our end users. And we are focusing on both retrospective and prospective verification of biomedical data. So today I will give you a very quick introduction to what biometrics is and why do we need FAIR in clinical studies. Then I will introduce the program itself, and then I will give you a couple of examples of how we are approaching this verification from both retrospective and prospective perspective. So biometrics is really a main driver behind verification of clinical data. And In biometrics, we are focusing on designing, capturing, and analyzing medical data that consists of clinical, biomarker, and genomics data. So it's a really wide set of different data types. The goal of personalized healthcare is to extract information from all of that data that is available either in current studies or most of all in the past studies, to bring the right medicines to the right patients at the right time based on their individual characteristics. And to achieve these goals, it is required to analyze huge amount of data across studies, but very often also across diseases. And it's virtually not possible without having access to FAIR data. So why do we want to use the data for the second time at all? We have a large amount of data across different studies that have been collected over the years for a very specific reason and to answer very specific scientific questions. And when a study has a negative outcome, very often this data is not reanalyzed because the study is closed. So we want to make sure that the data that is collected from our patients is not lost and can be used to their benefits. Of course, in accordance to the consent that they are providing to us, but it really creates an opportunity to revise the assumptions and linking it with other data types, not necessarily those that have been linked before in set of clinical study, because some of those data types are collected for exploratory reasons. So the second use of data may apply to either one study or multiple studies in one program or one product as per the picture, or even across the programs. Of course, it poses some challenges. The most important ones are listed here. So we are dealing with different standards. I will be talking about that a bit later. We have quite complex data structures. As I mentioned, we have different data types, genomics, imaging, biomarker, and different IT systems. And those IT systems come with different data types, but also in different phases of those clinical studies. So I will be not talking much about FAIR for the sake of time, but if you are interested in more details, I really recommend you to get familiar with FAIR guiding principle for scientific data management and stewardship that has been published in 2016 in Scientific Data. And it really describes in more details what does it mean to have definable, accessible, interoperable, and reusable data. What we have decided to add in Roche is also the shared part, because for us it is important that whatever we are doing to verify the data, it can be shared. And here we are not talking only about those integrated data assets, but also the insights that we are generating as a result of it. So Roche vision is to accelerate insights generation through better data management. And to achieve this, we need to change the way that we work to enable faster and more effective use of our data. To do that, we have to work on various elements like data management, single point of truth, or how we call it, harmonized data access point. And you can hear a bit more about that later. Data integration, and also the mechanism that we apply to share both the data and the insights. And this is done in order to feed the forward translation and reverse translation. So if you are not familiar with those terms, forward translation is what we are doing in the setting of a clinical study, whereas reverse translation is when we take those data from the past studies and the learnings from them, and we inform the research for further insights and hopefully later drug development. So ED stands for Enhanced Data and Insight Sharing, and it aims to enable management, integration, and sharing of biomedical patient data. We are doing that to accelerate the generation of new insights and to advance drug development and discovery. It is not a system. It's really a set of or pool of people, processes, and tools and systems that we are creating. When we started the program, we focused on people. We hired curators and integrators that were supposed to start generating those data marts in a quiet, manual way. in order to identify the problems and find the learnings and opportunities that we can apply further in the process. So when we had this experience, we started working out the sustainable processes that then we can actually inform creation of those tools and systems, or as we call it, EDIS end-to-end engine, which consists of technologies and processes supporting prospective and retrospective verification. And while building this engine, we are using so-called NIH Data Commons model, which refers to information, data, and content that is collectively owned and managed by a community of users. And it creates kind of a flexible and fit-for-purpose framework. So again, it's not a system, it's not a specific technology. It's a way to holistically manage our data and our metadata by storing them, accessing, and sharing in a way that gives us flexibility. So again, we don't have one storage for all data types or all kinds of users. We don't have one way of analyzing this data for all users. It's really fit for purpose. And it starts with the raw data, and it goes through the integrated data sets and visualized insights. And apart from those 5 layers, it also uses number of supporting services, like, for example, reference data services, which apply to metadata, to terminologies. It provides APIs to access all those data across the layers, and of course, authentication and access management for users to have access to this data and be able to share it wider with the organization. So with that retrospective verification, why are we doing that at all? We have a vast collection of diverse data that provides us the link between patients and our ability to develop medicines. It provides a very unique opportunity to create drugs beyond anything that one function or one research program can generate. So in EDIS, we are investing significant amount of time and resources to find and access and harmonize and integrate the data from the past studies in order to create the data marts to extract even more value from these legacy assets. So what is integrated data mart? In each study, we have data from different sources, different types, and all of that can be linked within this particular study. And when we combine these integrated studies in a harmonized way, we actually enable our scientists to compare the data from one study to another, from one program to another, sometimes even across different diseases or therapeutic areas. So what is the process of this retrospective verification? At the beginning, we start with defining the scope. By that, I mean trying to understand which studies we should focus on and which data types are important. Later on, we are going also to what data points in those studies is valid for us, because maybe we don't need to focus on all data that has been generated in the past. Then we curate that data in order to bring it to the same format. I will be talking about that process a bit later. We integrate clinical data by creating so-called ADAM datasets or analysis datasets. Then we integrate it across domain, so across all those different data types. And then we have the integrated data mart that is published and cataloged in a way that it is findable. So what is curation? I will give you a simple example. Imagine that you have 2 telephone books, and both of these books use different standards, or maybe no standards at all. So you can see that both of them have different variable names. They have different details. So in one case, you have the whole name in one column. In another, you have 2 columns for the first and last name. You have different standards to provide information about gender, different formats of date of birth. Sometimes, of course, you may have missing information, but as you can see, it is very difficult to read it that way. Even for a human, it is impossible to analyze such data in a machine format. So as part of curation, we are deciding on the data model, data standards and terminologies that we should follow, and we'll bring all of that from all the sources to this one format. So it is really easy to read and understand this data. So what is the current curation workflow at Roche? I will give an example of clinical data curation because it may differ from one data type to another. We are taking data from clinical systems, or in some cases also external data. And then we are following the process. We are using also some metadata and terminologies and data standards from reference services in order to inform this process. So first we access and pull of that data, and by pulling I mean bringing all studies in scope together, all data sets that are in scope together. Then we map and transform them in the way that I described on the previous slide, bringing all of them to the same standard and applying the same terminology. Once we have this data set, harmonized data set created, we annotate it, document, and catalog, so it can be findable, not only in context of the integration, but also in the future if we want to reuse these harmonized assets again. And then we are applying various quality control measures. When such data set is ready, we store it in clinical data repository, which we are creating also as part of EV's program. And we are documenting it in the metadata catalog and in the document repository wherever it is applicable. Of course, there's a number of challenges that come with the curation process. So first, it is actually being able to find and access the data, especially for past studies. They may live in different legacy systems. It may take time to verify where and which version of this data is usable for us, which should we use, and then how we can get access to this data and with whom should we talk about that. Something that we had to also solve at the beginning is which tool to use for this curation. At the moment, we are already behind this, but it was important discussion that we were having over first 1 or so years. And then the standard that I mentioned before. So data from different studies can come in different standards because they were developed in different years. So maybe there were no standard at all. Maybe it was actually a different standard that was obligatory at the time. Some data may not be standardized at all for various reasons, or study has could apply some study-specific customization. So all of that require significant data expertise. Then the standards can change over time, and that applies not only to that source data, but also how we manage those harmonized assets to make sure that they are up to date and in current standard when, for example, SDTM standard may change twice a year. We are dealing with quite a large amount of data and different data types, different domains. That this also poses challenge for the tools, for the storage, but also for human and the process itself. So now the integration part. Why do we need to integrate the data at all? I will start with a very brief example. So imagine that there is a drug and some patients after taking this drug develop quite severe skin rash. So the patient goes back to the doctor and the doctor thinks that, okay, the skin rash is so severe that they will take the patient off the drug. But then the further analysis of outcomes in patients that continued to take this drug, even with the skin rash, shows that these patients respond to the drug much better. So, for example, they may have higher overall survival, or they actually are treated much faster than the others. So if we connect all those data, the clinical data and the biomarker data, genomics data, we may find a biomarker that show us that a specific patient will develop this skin rash. And by that, we can inform doctor and the patient that for this particular patient, they will have the skin rash, but please treat it. Please don't take the patient off the drug, because they will actually have better chances to get treated. So as you can see, discovering such links provide huge benefits, and it helps us to personalize drugs to meet real patients' needs, because we are far beyond targeting one drug to the whole population. So what is the integration process that we are following? First, we need to understand the use case and the scientific questions that are behind this use case in order to decide on the scope of the data. So here, the studies, of course, that I mentioned before, but also what data types, what kind of biomarkers do we need, for example, RNA sequence, or do we need the whole genome sequence that needs to be linked together? Once we have this formulated, then we are extracting this data from this harmonized assets, harmonized sources. We apply predefined model, and then we connect all this data using mapping methods that we have developed. Once the data is linked together, we produce the integrated data mart that is then shared with anyone who is interested in this, particularly the use case team that might have requested it at the first point, and we provide access to it from various visual and analytical tools. So again, it comes with a number of challenges. One is size of data. So if we were talking about size of data in context of curation, now we are taking multiple data types, very often really significant amounts, like a whole genome sequence, together, and we need to make sure that it can be processed in a reasonable time, and it can be stored and accessed without a huge impact on the performance and ability to analyze this data. Then understanding the requirements and the scope for derivations. It also takes some time working with scientists and the use case teams. Of course, at the beginning, we also had to solve the problem of which tools to use, but most important is in what format to provide this data, because we may choose between formats that enrich the data in a significant way by providing different linkage, different metadata. But on the other hand, we have to focus on the data types that are easier to use by our end users, so it actually can be used by a wider audience. Then linking samples to patients, this is quite difficult process at the moment. Data mart versioning, so what if we want to add additional patients or additional data types or additional sources? That is connected highly with the anonymization to meet GDPR and other policies. Because if we have anonymized data, how can we ensure expansion of the data mart? So if we have anonymized patient ID, how can we then enrich this data mart with another source of biomarkers, as an example? And then how do we share those data marts, and how we ensure that the right people have access to it? Because not all data can be shared widely across the organization. And that brings us to prospective verification. So it involves applying the FAIR principles, practices, and solutions as early in the process as possible. So already when planning the study and when acquiring the data. And it really means that we collect the right data in the right way. And by data, I mean both content and context. So we need to make sure that we have the metadata, we understand what standards have been applied, what terminologies have been applied, and so on. And to meet those requirements, we are creating so-called EDIS end-to-end engine, which spans across all those 5 layers. And it's a set of processes, tools, and systems to support the study teams along this FAIR data journey. I will give an example of the very last or almost last piece of that engine, which I'm personally involved in, which is a data integration, sharing, and exploration platform. It's a component of the EDIS end-to-end engine that goes to layer 4, which is integrated data assets, and a bit to layer 5, which are the insight generation. And it helps data integrators to get the data from the harmonized data sources and create those data marts, link those different data types first, and then share it with scientists, with analysts, for them to access and explore those diverse types of data, here specifically clinical and biomarker, but also by providing link to whole genome sequence and others that may be stored externally, different system. And it enables solving complex scientific questions by conducting queries in a dedicated dashboards that are part of the Pulsar platform, or by exporting this data to external tools for more advanced analysis. But even the best set of tools and processes won't be successful without the right mindset. And for that, we have created so-called FAIR playbook, which provides an overview of shared rules from management, for management of internal data assets, from before it is even ingested to our systems to the time when it is ready for the secondary use. Three major components of this playbook is data citizenship, when we, as Roche and as every employee, are collectively responsible and accountable for the data that we either create or consume or manage and share. And it's really important to shift this mindset from it is my data to it is our data. Internal data access principles provide very important guideline on how do we need to behave, either as a data provider or as a data consumer, and how do we want to work together to ensure that we are bringing the benefits to the patients. And then the FAIR data management that goes along the EDIS end-to-end engine and is really part of every step from the data planning to reuse. And with that, I would like to thank you very much. We will have a chance to talk again in 30 minutes. If you will have some questions at the later point, then please free to contact me either via email or via LinkedIn. Thank you very much.
Okay, thanks very much, Ewa. So don't hesitate, everyone, to put some questions already now in the chat, so then we can collect them for the end. So with this, I would like to hand over to our second speaker, Patrick. I think you should be set up to share.
Okay. Do you hear me at least?
Yes, we can even see you. Okay.
Okay. So I'm going to open up my presentation. It's here. I will go in presentation mode, but just confirm that the slides moves on the screen.
So
you
far,
can see the screen, yes.
possible, yeah.
Okay. Okay. So thank you everyone. Thank you Daniel. Thank you Wega for the invitation. So I'm going to talk about the very same topics, which is FAIR, data management, but let's say more from the point of view of an academic perspective. I'm going to talk about Swiss EGA, which stands for the Swiss Enhanced Genotype Phenotype Archive. So the name is borrowed from the EGA. I don't know if you're familiar with the EGA. It's an archive which is maintained by the EBI in Cambridge, and it stores data which contain some clinical information and some omics information. Okay, this is supposed to cover all research in Europe, and there are other similar archives in the US like dbGaP and in other places. Okay, so Swiss EGA will be, let's say, the local EGA for Switzerland. So how all this started? So there are several stories, but maybe one of the tipping point is that we understood a couple of years ago with the GDPR coming that we need to articulate the legal together with the constraint related to the publication of research data. You know, this is typically FAIR. So we are sort of squeezed between the fact that the data needs nice management and stewardship, with the GDPR.
Yep. Is it intended that you are on slide 21 or?
Yeah. This is what I was fearing. So actually, I will have to go back to the. I will have to go out of the slideshow because I think my slides do not move otherwise. Okay.
Yeah.
So here you should see different slides now. Am I right?
Yes. We're on slide 2 now. Yes.
Slide 1.
Yes. It's a bit slow.
Yeah, it's a bit slow. So, okay, so it's something I already observed with Teams. For some strange reasons, the slideshow does not work always very, very well. Okay, so now you should be on slide 2 in principle. And I was just referring to this tweet of mine a few years ago, where we had a sort of conflict between the GDPR, where we occasionally need to delete the data and to handle the data in a way which is consistent with the participants and the patients, and the fact that for scientific FAIR data management, we need to archive the data so that data can be reproduced. Okay, so in a sense, there is a sort of conflict here. So still, the conflict can be solved. So I'm going to talk about the solution that we are trying to implement. So some background information. I'm going to provide the big vision of the data lifecycle management. I'm going to specifically discuss Swiss EGA and in particular the so the findability of the data within Swiss EGA. I will provide you with a pilot study that we perform and some conclusion and take home message. So what is what is the origin of this, together with this let's say Twitter discussion, is the fact that in Switzerland we engage into a relatively ambitious project, which is the Swiss Personalized Health Network. It's well-funded project from the SERI, okay, so and it's coordinated by the Swiss Academy of Medical Sciences. Within that particular program, which tries to build an infrastructure to manage personalized health, we have a specific earmark funding, which is managed by the Swiss Institute of Bioinformatics, where we try to provide the research infrastructure to manage the data. It means secure data, but also all the life cycle of the data. So I guess you are familiar with the with the FAIR, where normally a study starts with a study design. Then you need to collect your data, and you need to collect them in a way which is compliant with the regulation and the consent. Then you need to do the data analysis, and then you generate some knowledge that you are going to report on, and in academic it means to publish. Okay. So this is the general framework. So the data, scientific data, must be shared, but they must be shared under specific conditions. So now if we dig a little bit more into this, let's say, circle, so you can start at the top of the circle. There you have you need to generate some data, so you have an approval from an ethical committee, or in a pharma it will be for maybe for a trial from an authority. And so you generate the data. Then you will have to manage this data that you generate. You may use the same tools like Pharma, RedCap, or LabKey, or whatever. Then you will do the analytics, and then we go down to the circle. At some point, the data, let's say, are kind of shareable. So you would store them in a specific vault. Okay, you may send your CDISC data to the FDA, and you may publish the results. And the publication channel will be probably the same whether you are in a pharma or in an academic. You may end up having the result of your trial or your study in PubMed and Medline. Okay, this is a very important time because there the data, in principle, could be reused by someone else. Okay. To make sure that the data can be reused by someone else at time of publication, then you need to build systems so that this data can be found. Okay, So this is the data exploration, step 6 on the slide. And occasionally you may jump from step 6 to step 1 because you just don't find any data that are relevant for the study that you want to perform. But if you find some data which are relevant, then you need to file a data request, which is going to be filtered by the data access committee. And again, you start the loop. You will maybe have a secondary usage of the data. Okay, so this is how it works in the academics, and it's not that different in an industry. So what is quite interesting within SPHN is that, of course, we have this this loop where we collect the data from the healthcare system, then we do the research work, but very often this is already happening in 3 years now that the program has been started. We influence the healthcare system. So, for instance, one example I can give within SPHN is that people doing research in oncology code data with ICD-03, okay, which is a version of ICD for the oncology. And today, hospitals start coding oncological data routinely via ICD-03, which was not the case 3 years ago. Okay, so in the sense, because the research is valued, in a sense, it's able to influence the clinical practice itself. Okay, so the rest of the circle, you are familiar with it. You know, we have this research which mostly would include, you know, imaging, omics data, and then the regular electronic health records with what you find it there, and you need to combine all this. I think the presentation from Ewa was excellent because maybe the focus of her presentation was on interoperability, which is not the case of my presentation. And then you can end up with predictive, preventive, or any kind of outcome. So for us, the problem to be solved. So we missed a platform to support the findability of the data. Okay, if you look for data sets, you have nowhere to go. And you need to look for data sets which cannot be fully public because the access must be monitored by some ethical committee and data access committee. Then, of course, there is always this issue with the consistency of the metadata. Okay, this was well identified. Yeah, in a sense, interoperability is really a grail here, but we should not forget that we also have to find the data sets. So we need an effective way to store, find, and request these data sets, and all this has to be compliant with the regulation. And I don't know, very recently we had this Lancet retraction paper with the University of Zurich. I guess you all heard about this. And one of the issue here is that the data controller, University of Zurich, never accessed the data. Okay, so now if we look at in practice what the Swiss EGA is doing, so we need to manage data types. So we have all kind of participant level data, you know, the same like the pharma, ICD, low-inc, and so on. Okay, SNOMED probably. Then we have all kind of omics data, pretty much the same. And then we have the interim reports, you know, all the analytics, statistical analysis, and so on. And then we have some images. So in general, most projects will generate relatively heterogeneous data. You know, some project will generate probably quite unique data. You know, if you are a project in lipidomics or glycomics, then there are even almost no standard there. You know, it's just, this is the way research works. They explore new areas, so don't expect to have strong standardization there. You know, it's new. Pretty much like Illumina was new several years ago. So if we look at the type of data types that we have today within SPHN, so we probably have 80% of the data that are possible in research. Okay, some of them are missing, like we do not have typically clinical trials data. We do not have this type, but we have interventional studies which are relatively similar. We have no cell signaling actually, but all the other types we have. So this list of type is borrowed from DataMed, which is the NIH dataset catalog repository. Actually, one type that we have within SPHN projects, which are not in DataMed, are narratives. So we have sizable content of clinical records, which are written simply in text. These are follow-up notes or discharge summaries and so on. And this is relatively, I think, unusual in a way. Okay, so then of course we have the archiving steps, because you have this big circle. Normally in archiving science, you have these 3 ages of the data. So you have current data, the data that you work with during the duration of the project, and then you have something which is after completion of the project. Completion plus a couple of years. This may depend on the data usage contract, and also it may depend on the specific regulation. Publishers would at least ask for probably 2 years, and some others may require longer time. And of course, trials data may be archived for 10 or 12 years. And then there are historical archives. And I think within SPHN, we cover relatively well the 2 first ages, but the last one is probably something still to be built. So I go on. So to recap, we want to provide the end users with a dataset registration system. And with the same system, you want to be able to discover existing datasets within this SPHN landscape. So we will cover datasets produced by SPHN, but probably also try to go beyond that, because the effort is significant. So you will have to provide guidelines to annotate properly the data. But again, as said before, to have, let's say, predefined standards to describe the data is a sort of impossible project in a sense. Research tend to generate its own standards. Okay, you need to provide an integrated system to expose the data and to search them. And of course, you need to facilitate the access, because once you locate the data sets that you like, then you still to provide the user with all the mechanism to put the ends on the data set. It means that you need to have within the loop, this data access committee and of course the logging and the secure access and all that type of infrastructure. So what we have today is a portal, so you can search for the data sets, and you can even visualize some of the data contents. We also have an international, let's say, branch of the project, and we work with EGA, okay, where the idea is to have different tiers. You will have maybe a Swiss tier of the EGA. We have many of the data will stay in Switzerland, sometimes for legal reasons, sometimes simply to share the burden. But at least the metadata describing the data sets could be pushed to EGA, or further than EGA could be pushed, for instance, to DataMed, or maybe tomorrow they could be pushed into Google dataset search. Google is engaged in very similar initiatives. Okay, so of course, to have a search, you need a sort of homogeneous index. So because again, the data sets are highly heterogeneous, we will rely on automatic systems to assign the descriptors, which then will be validated by a human within the loop. Okay, so we have, as usual in a data warehouse or data mart, we have the physical model, and we will use DATS and JSON. We have a logical model, so we intensively use linked open data framework, an RDF and the like. And we have a semantic model to describe the content of the data, and we will rely on the broadest coverage vocabulary, including UMLS and OBO and the like. Interestingly, when you look at the semantic model, you need to separate the description of the data, I mean, what the data is about, and the administrative aspect of the data, under which condition the data can be shared, how long can you store the data, and so on. So where we stand today is that we have a data ingestion framework, which is able to let's say, harvest virtually any type of content. We even are testing the extraction of content within images. Of course, I will provide an evaluation of this. So we are able to annotate and index any type of content. We also had pre-existing work with actually that we made together with Novartis on the processing of informed consent, which was relatively successful. We were able to extract, most salient information like sample and storage duration with a precision higher than 98% into a dozen of languages. So with this harvester, we claim that we can, let's say, with brute force approach, capture the most salient information within a data set. And for the rest, we will rely on the human in the loop. Now, if you look at the data set exploration, which is the probably the endpoint of the initiative, we use the BioCADI benchmark to evaluate the search effectiveness of the system. So the benchmark is publicly available. It has been developed by the NIH. And the typical question are the following. So you want to search for data on a particular disease or different disease that mention a specific tumor type and a somatic variant. Then you can look for studies about a particular biological process, for instance, obesity as measured by body mass index, and with different intervention, including surgery intervention or anything like that. In Switzerland, it will be coded with SHOP or ICD-9. Then you want to filter by demographic characteristics, age-related groups, gender, anything like that, ethnics. And then third case, you want to find cases like a particular case. So you have a certain patient, he has some treatments, he has some specific response to some treatments, and then he has specific germline variants, and you want similar cases. Okay, so this is all covered by about 50 questions within the BioCADI benchmark. So immediately I can provide you with the results. So that was published in 2017. We were several competitors, including commercial players, and so we achieved very effective results compared to the others. Okay, so we have a very state-of-the-art system. And we try to use very fancy, deep neural networks, query expansion and normalization. But the most effective approach are very, let's say, straightforward. We use, I don't want to go into technicalities, but we use, search engines like Lucene and Okapi BM25 weighting schema and so on and so on. So this was a bit disappointing, but I think search engine, the regular search engine are able to do the job very well. And so far, the let's say most advanced search and analytics are not so effective. So okay, we were very proud of us. We are happy. But if you look at further into the detail, actually these numbers that we get here, which is about 30% of precision for the top 10, it's relatively good compared to the others, but it's relatively bad. When you search content into Google or into PubMed, the precision of your search in the top 10 results is closer to 70 or 80%. So 30% isn't good, which means that probably findability, which is really probably the basis of fairness, still need to be improved. Okay, so in conclusion, we are relatively happy with the search effectiveness of the tool that we developed. So this is clearly state of the art. We were a bit disappointed in the sense that deep or machine learning has yet very little impact. We probably have some, we have some ideas why it is so and why it may change in the future. So the room for improvement is huge. As I said, the baseline for, let's say, general search engine is probably 80% in the top 10 results. And we are really, really far from it. So which means there is still room for research. So if we try to go a bit further, let's say, to do some analogy with what we do in the academics and what would happen in some in Novartis or in Roche or in these companies. So I think that probably the data sharing policy in pharma could be, let's say, revisited. So many of these companies are engaged in building enterprise search engines. And I think this is a very good move because one of the challenge when you work in these companies is already to locate the data, okay? And I would say that these companies are today investing massively into machine learning. Some move to Barcelona and let's say they took it seriously, but in a sense, search technologies have been relatively neglected. The second thing is that data set registration should become routine everywhere. I would say that it's kind of complementary to the data mart and silo logic, which is probably dominant today in this business. But the data set has many advantages in the sense that it provides, a very consistent access to the data. You don't need to pick up the data into a dozen of silos, so that it's something that could be complement to the data mart approach. And then for the semantic interoperability, which is still a huge issue, I think that while for the descriptive metadata, we have standards developing. For the administrative metadata, how the data can be shared, under which condition, what is the legal, for how many years. Although there are some initiatives like the data use ontology, I think more effort is needed. And it's more effort is needed on the semantic interoperability, but also maybe more generally in the linking of this data with the research data sets. Okay, I think I'm done with my presentation. Thank you very much. Thank you, Daniel. Thank you, Christian. And thank you for listening to my presentation.
So thank you very much, Patrick. And also thanks again, Ewa, for these excellent presentations. So I think there will be some questions. I saw that one question from Aurélien has already been answered. Do you want to say a word about it for the rest of the audience?
Sure. So there was a question on how we do ensure that all the informed consent allow for a use of the data in different contexts. So there are different approaches to different situations. In the past, a lot of consents allowed for a specific use within a clinical study, but then we always had to verify if a specific consent for a specific patient allow us to use data that we are interested in, also in other contexts, so either secondary use or exploration and so on. Going forward, we are actually working on consent files, so they address those needs already. So patients can contest to our exploratory or secondary use, and that way we can then verify this in a more automated way in the future. At the moment, very often it's one by one for each patient and each data type analysis.
Okay. Thanks. So there is a question for both speakers from Graham. I think clinical study data needs to be available forever. Would you agree to that?
So as far as I know, and I don't remember exact number of years, they don't need to be available forever, although it is significant amount of time anyway. So at the moment we have vast number of past studies that live in our systems for 20 years and more.
Yeah, maybe I can also try to handle that one. So I think so eternity is long, you know. So so the only thing I can try to answer is that very often actually we underestimate the cost of this long-term data management. So as soon as you come up with a statement, you probably need to price the cost of this statement. And that is true everywhere. That is true in pharma. That is true in the academics. And I would say that very often in the academics, the funding agencies have not been ready to pay for these costs. You know, you normally get a grant for, you know, whatever, could be a few months, it could be a few years, it could be some grants can be very long, but they rarely commit forever. You know, that's probably part of the challenge that we have addressing this issue. The other aspect is, of course, is that you have the legal aspect. So the legal aspect anyway is constraining you. So you normally have minimal duration. So occasionally you have maximal duration, and sometimes you have anything in between. Okay, so that's where we stand.
But if we take the question the other way around, is there a notion of obsolescence of some data because at some point they were based on wrong assumptions or on not well-known assumptions of whatever biological process? Do you think it exists, or?
Maybe I don't know. I can also try to answer that one. It depends on how do you find the obsolescence, in a sense that from a scientific point of view, I would argue that there is an obsolescence of the data. From if you consider now, you know, other aspects, you know, patrimonial or history of science, you know, I mean, where do you put the borders, you know? If you consider things in the long run, you are very happy to be able to study how, you know, if you do research in epistemology or, I mean, everything, all these things that I know nothing about, but still, you could imagine that these guys need very, very long-term archive, okay? And that's a different usage than to look at reproducibility of the data, for instance, okay? Yeah.
And I would also add that, yes, sometimes we don't even want to reanalyze very old studies because they might have been conducted in context of different standard of care and something that we are not interested to follow anymore. Mm.
Okay, very good. Thanks. Then I have another question from Saravanan for Ewa. Can you please advise on the proportion of in-house developed solutions and commercial off-the-shelf solutions in this program? I understand the integration probably happened in-house, but I'm thinking of the individual components.
So the specific proportion may be difficult, but we have definitely a mix of developed solutions as well as commercial off-the-shelf solutions. We are trying to address those needs in a component-based approach. So instead of having one fit-for-all solution that usually do not work. We have set of tools. We are using also such popular tools like R or Python, especially for the curation and integration. Those are very important tools. And then we are trying to address a specific need with a component that addresses it in the best way. So we have mix of off-the-shelf solutions, sometimes customized to our needs, and in-house development.
Thank you.
Okay, thanks. There is a question from Faye.
Actually, the next two questions are for Patrick and kind of related. One is, how do you register data sets for non-IT people? And the second one is about the current size of the Swiss EGA.
Okay, so how do you register data sets for non-IT people? So normally the GUI that we have are relatively let's say user-friendly. So you can you will simply log in and then upload your data set. So this is, I think, relatively managed, and I think similar projects are doing the same. So dbGaP, EGA has pretty much the same type of interface. So it's not, there is no room for a demo, but basically you don't need to be an IT to do that. The other aspect, the second question was, what was it again?
Is the size, the current size, number of studies, and
okay. So we've been
data.
Yeah. So I can say that I think today for the data sets that we have, it's you know, a handful, you know, like maybe half a dozen or a dozen of these data sets are within Swiss EGA. So this is because simply the program started in 2017, and the project are only today generating the data that are likely to be shared. So the sample that we let's say have to handle are relatively limited. But the tests that we made within with the BioKD, I mean, let's say the scalability test that we did with the BioKD benchmarks was close to 1 million data sets, you know. So it's sizable. Yeah.
Yeah. And some precisions around the type of associated data, metadata, which you currently have, and the rate at which it grows.
So again, the rate is maybe not significant, because in a sense, when you have so you have like maybe 20 projects which are active, then you have 6 of them that are already in position to release data. If one of them, the number 6, release enormous omics data, so the storage explodes, you know. It's already 1 terabyte, and then you have a growth of a factor of 1,000, just because the one before were releasing, you know, simply clinical data and variants, you know. So the storage is not very significant here. So I cannot. I think talking about the growth is not really is not very useful. But the data types, I have this slide that I showed. So it's really almost everything you could have. You know, we even have, as I said, medical images. We have nucleotide sequences. We have a lot of phenotypes. You know, any type of phenotype diagnosis. Labs, we have sensor data. Of course, we have all type of proteomics. We have registry data, epidemiology data, registry data, you know, like cancer registry data. We have physiological signals and, you know, sequences, you know, cardiac sequences, all type of this signal stuff. We have epigenetic data. We have data from papers, you know, like meta-analysis of papers. We have narratives, for instance, from radiology reports. So we have radiology reports that we try to classify into RECIST. You know, RECIST is this where you look at whether the response to the treatment is positive, negative, or stable, and so on. This is very familiar for the trials data. Okay, we have virtually all type of data. So I don't know if a particular type of data is important, but yeah, it's very broad.
Does it answer your question?
Yeah, it does. Thanks for that.
Thank you. Another question from Faye, and then I may have personally some questions, but I want to let the people ask the questions. Another thing, recently I've been looking for metagenomic studies. Related metadata and pre-analytical data were totally absent. Any idea why or how? Any idea? Oh, sorry. Any idea why those pre-analytical data? Any idea how those pre-analytical data could also be included, since they're quite critical for the analysis of the results?
Well, I guess it's also a question for Ewa. So I guess so. One case where we pay a lot of attention to this pre-analytical data are on the for the management of biobanks. So like pharma, actually, we manage cohort. Okay, but probably in the academics, the cohort that we manage can be managed over a very long period of time. You know, we manage cohort for 10 or 20 years. You know, so there is a long follow-up of the cohort. And we know that when we had surprises, you know, you collect your sample data, and then you have huge differences between the sites where you collect your data. And actually, it's usually the difference are due to different pre-analytical, you know, aspect. Time to the fridge, you know, yeah, like it could be a few minutes or a few hours, and then it changed totally the concentration into the sample. So we address the pre-analytical issues very carefully in general for samples, which actually could be also valid for metagenomics. Definitely, this is very important. We do not have much. We do not have much metagenomics or microbiome in general within SPHN today. So maybe Ewa can continue on this.
Yeah. So just before you continue, Ewa, can you also both address at the same time the access to the samples? Is that something you think of related to the studies? And that's for both of you too. Sorry, Ewa.
Finish on the previous answer. We don't have metagenomic studies at all. It's not really a clinical setting, but it would be probably dependent of where did you access this data and what exactly the data was. I could think of the size of this preclinical data as one of the issues that could lead to the fact that only analysis results have been shared. And regarding the access to the samples, I think I would need more context for your question.
The question is, if you make all of those data sets available, and there is an analysis made out of it, which shows that it would be relevant to get access to some of those samples to do some further analysis, is that something you try to envision in that global picture, or is that just already so difficult to get data that the samples will come next?
No, of course, if we are talking about the access to the data, we are talking about access to both integrated and the source data. So, of course, as part of the curation integration process, we do need to have access to the samples, for example, to ensure that the constants are there, and also that we are linking the samples to the patients. And then in those integrated data assets, we actually have those specific samples. So you can do an analysis for a specific patient or for a specific sample.
Yeah, okay. On my side, I would say that within the data set catalog, actually we have place orders to cross-reference other repositories. So one of them is obvious, is the publication. So we have a placeholder, you know, for DOI and PubMed and stuff like that. That's obvious. And one of the placeholder is for the biobank itself. So we will directly link within the data set catalog to the Swiss biobank where the sample is stored. Yeah, exactly. Yeah.
Okay. A question from, I don't know how much time we have left, Christian.
Yeah, we have a few minutes. I think there is another question from Zoe for Patrick. Did you have any problems with analyzing doctors' notes or free text with NLP, considering the different languages used in Switzerland?
Yeah, so actually, we've been one of the projects, so this Swiss personalized oncology project, which gather virtually all oncology hospitals in Switzerland, because we have the 5 university hospitals, and via the SAKK we also have Hospital Arrao, or we have St. Gallen. We have so we really cover all the oncology in hospital, okay, not in a private practice. And actually the narrative themselves, we handle them in French and in German, okay. Well, a marginal part of them in some canton like Geneva is in English, but it's marginal, okay. So it's mostly German and French, obviously. In general, the radiology report, I mean, I know what the question referred to. You know, some follow-up notes may have some weird abbreviation, and that is absolutely true. The tool that we have today, and depending on what you want to do, okay, the use case here is take a radiology report, assign one of the 5 RECIST codes, you know, disease progression, disease regression, stability. It's relatively simple. The approach that we use are relatively language independent and relatively also robust towards corruption that may occur, you know, misspellings and stuff like that, you know. So we basically train a model, we cross-validate, and whether we get 80 or 90% precision, then we can work with this, you know. So no, I would say no. So definitely there may be other application where this type of let's say linguistic challenge may play a role. I would say it's in general you can overcome this kind of issue. Yeah. Okay.
Do we have questions? No, but I have.
Yep.
Oh, yes, there is one. Why is that? Question
Okay.
based
I'll
on
take that. Oh, go ahead, Daniel.
No, sorry. Yeah. Based on the example of COVID-19, did you make changes to your approaches? It seems that all companies, researchers were desperately looking for past data.
Yeah.
So,
yes, of course. So, as part of the program, maybe the approach specifically did not change, but we have created a data mart that is relevant to the COVID-19. And it was done much faster than the previous data marts. But one of the reasons is that we already had all those processes and tools in place, at least those that are critical. So we did not have to develop everything from scratch. It was much easier to do to find the right data and do the analysis because of what has been developed before.
Patrick?
Yeah, I can. So I would say the impact itself is probably relatively marginal on the design of the system. But if I try to pick at least 2 things I observe is that we probably will have to link not only with the publication but also with preprints. This is getting obvious, you know, especially for COVID-19. You know, something like 80% of COVID-19 publications was in preprint, okay, and it has advantages and it has a few drawbacks as well. But this is something that we clearly take into account now. The other thing is that probably if you look at the semantic interoperability, I would say that it's very interesting in the sense that you will have more projects in the coming years. Paying attention to the biodiversity. So, of course, we will continue working on viruses, we will continue working on comorbidities, we will continue working on drug repurposing. What is probably new is that there will be a focus on this, how could I call it? Yes, this specific zoonosis in general, you know. But also the border between animal and human separation, you know, will probably get a little bit more fuzzy than it used to. Yeah, this is what I expect.
Okay, thanks. Daniel, do you have a last question?
Yeah, maybe just two last questions, and it's kind of coming back to the introduction. Well, the question is twofold. What sort of strategies are you employing to promote the work you do and the capabilities you provide? That's the first side of the question. And the second is, do you already have, or is that too early, do you already have measures of success? You know, can we, which we could relate to those 10 billions, which was stated in this tweet, or is that too early?
Yeah, so I think on how we are promoting it, I was trying to reply to that question a bit in my mindset slide. So we are having significant efforts to promote this new way of working and new mindset around sharing the data and ownership of the data. Regarding how we are measuring the success, as I mentioned, we have different phases in the program. So first we tried to focus on people, then processes, and now on the tools. And we have our goals around those phases. So for example, we had goals and KPIs around patient data that has been integrated. So as an example, one of the goals was to have 35,000 patients worth of data curated and integrated across different diseases and therapeutic areas, which actually turned out to be, after the whole year, 180,000. So we are exceeded that goal quite significantly. Now, for example, we have a goal around prospective verification, where we are looking at number of studies that are applying those new principles and new ways of working from the planning stage to ensure that this prospective verification flies. And of course, this is also a very good testing bed for us to further improve the engine.
Okay, Patrick.
Yeah, so your question raised the question of the, let's say, success criteria in a sense. So we of course we have the usual success criteria, you know, the outputs, you know, how many publications, blah 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 blah. But this is I think injecting, you know, 100 million or 200 million within the ecosystem of research in Switzerland is relatively marginal. You know, research in Switzerland is probably 2 billion, you know. So in a sense, what is more interesting is probably to look at the specific objective of SPHN. So one thing I can observe is that in the usual process to access clinical data, so you are a researcher and you want to access clinical data, you need to go through the ethical committee and so on and so on, and it could easily require a year to get your hands on the data. You know, of course there are many differences, and we are talking about 20 projects. You know, so from a statistical point of view, the statistical power is limited. But so what I think we achieve, we are able probably to move from something to access the data, which could be a year, to probably something closer to a couple of months, maybe, you know, 3 months. So this is significant. It's significant. It's still relatively qualitative, so I cannot tell you that the improvement is with a p-value and so on. But we shorten the path to get access to the clinical data from hospitals. That is very clear. The other things, which is a sort of, let's say, almost a side effect, or a nice side effect of the program, is that this is something I mentioned, is that we managed to change the healthcare practices. So this is going somehow beyond what we were expecting, but we knew that the routine healthcare system is not necessarily capturing what is needed to perform research. You know, we know that the capturing of the diagnoses is geared toward billing. You know, this is well known. Yeah. I mean, it's part of it's one of the let's say bias within the healthcare system. It's a known bias, well documented already in the 80s and 90s. So we know we have this type of issue, and I think because we engage into this large program that also involve directly the healthcare institutions, we can change the practice. We can ask them to, of course, under a certain condition, to code things natively into ICD-03 or NCI Thesaurus or whatever you may need. And this is a huge change. This is a really, really enormous change. Yeah.
Great. Christian, do we let that last question from Yannick?
Yeah, I mean, we have 1 minute left. I'm sure we can take this question as well. Patrick, you mentioned that the performance of the internal search engines is far lower than the generic search engines. So what's the reason for that? Is it algorithms, or has the unsupervised training lower experience? And you mentioned that you identified ways to improve from the 30%. So what are these approaches?
Yeah. So to make a long story short, the substrate of the search. So we compare between dataset search and PubMed or Google search. It is clear that the material of the search is very different. So this is one reason why it's so we do not have such a success. So in one case you look for documents, and search engines are very good at processing text. In the other case, you look for Excel sheets, content. You look for anything, files, CSV and so on. So obviously, it is expected that it should be a bit more challenging. Now, still, it's very different. So what could be improved is actually, it's like a zip for Pareto distribution, So what we can do is we look at the type of data sets that we have. So we have a lot of, variant-related data. So, gene panels, genome, and so on. Okay, we have a lot of this. So we can build specific data handlers for this. Then some data are rare. I mentioned lipidomics. So maybe we will give up working on lipidomics, and we will not concentrate on this, and we will build specific data handlers for CRF, for this type of data, and this is the way forward. So pretty much like we are system to process PDF or system to process JATS or XML, we are going to do the same with we will have a cutoff. Say, okay, 80% of the data is this type of data, BAMF or whatever. And we will focus on specific data handlers for this. Then I hope I answer your question.
Thank you, Chris. Thank you, Patrick. Sorry. Yeah. Well, no, Christian. Before you close the session, I just want to warmly thank you both again. That's really a pleasure to listen to your talks, and the question session after was really great. So thanks again. And Christian, we're waiting for the croissant now.
Yeah, thanks as well. A big thank from my side for Ewa, for Patrick. So it was really great to have these two perspectives on the topic, and the lively discussions was also great. Thanks also for the guys who still hang on with us for so long. So some have dropped off, and maybe they hope for the recording. So we'll certainly provide that. So thanks again, and have a great day. Bye.
Thank you very much. Bye-bye.
Thank you.
Bye. Thanks. Bye.